Hello. Today we are going to talk about the basics of cytology. This is a very long unit. It's broad. That's the reason why we are going to subdivide it into different parts or sections. Now, to begin, we should know that cytology is an extremely important branch of biology because it deals with cells. All organisms are composed of cells, and all vital activities occur in cells. Cells are units of structure and function for living organisms. Cells are units of structure simply because they used to build up all living organisms. All living organisms in 5 kingdoms we know: Monera, Protista, Planta, Animalia, and Fungi. All of them are made of cells. So cells are building blocks for all living organisms without any exception. Cells are also units of function for living organisms because all metabolic activities, either anabolic or catabolic, important to sustain life, they take place in cells. So cells are units of function for living organisms too. Some organisms are unicellular. They consist of a single cell, too small to see with the unaided eye. Others are pluricellular or multicellular. They are composed of many specialized cells. When we say uni, it means one. The opposite is pluri or multi, meaning many. So this will help you to remember easily the difference between unicellular and multicellular organisms. Historical background of cytology. There is a very long list of scientists or cytologists who have tried to carry out experiments about the study of cells. The list is extremely long. The reason why I have chosen only 3 names: Robert Hooke, who discovered the microscope; Matthias Schleiden, whose experiments have shown that all plants are made of cells; and Theodor Schwann, the one who has shown that all animal tissues, or simply animals, are made of cells too. The cell theory. The cell theory includes the following 3 principles. 1. Cells are the smallest living things, obviously with some exceptions, because eggs of birds and reptiles are cells that are big enough to be seen with our naked eyes. 2nd, all organisms are composed of 1 or more cells. Already we know that all living organisms in all 5 kingdoms can be subdivided into 2 categories in terms of number of cells that make them up. Then we have unicellular organisms with 1 cell and pluri or multicellular organisms with more or many cells. 3rd, cells arise only by division of a previously existing cell. This element is highly important because it rejects completely what evolutionists used to call the spontaneous generation theory. Spontaneous generation theory. This was a theory that was used when people were trying to explain the origin of life, stating that maybe in some circumstances inert matter may give rise to a living thing. Examples were given like mud, giving rise to aquatic organisms like frogs and fish, like decaying cadaver or dead body, giving rise to maggots, etc. Maybe some of you may know some other examples of this kind. Cell structure. Normally, all living organisms are made of cells, but all cells are not alike. Cells differ in terms of size, in terms of shape, in terms of function too. So cells in living organisms are going to differ. They are going to present different characteristics according to the functions they have to carry out in living organisms. But at least all of them, they have some common major features, like a nucleoid or a nucleus. Remember that when we talk about the nucleoid here, you should think about the prokaryotic cells, a nucleus in eukaryotic cells. It is in the nucleoid or nucleus where the genetic material or DNA is located. There is also the cytoplasm, ribosomes, and plasma membrane. Ribosomes should be found in all cells, in all kingdoms, because as you know, ribosomes assume the synthesis of proteins, and proteins are substances of first biological importance, where for example in our body, proteins can be used as hormones, as antibodies, as enzymes, respiratory pigments, etc., etc. Let us begin with the plasma membrane. The plasma membrane or cell surface membrane, known also under different terms like cytoplasmic membrane or cell membrane, it surrounds all cells without any exception. Now, what's about the structure of plasma membranes? The plasma membrane is mainly made of phospholipid molecules, which make up a double layer or a bilayer, as it has been discussed in previous units. These phospholipids are supported by proteins, cholesterol, etc., etc., but mainly plasma membranes and all other membranes that we discuss about, okay, which can be found within the cytoplasm. All membranes in living organisms are mainly made of phospholipids. Now, what is the mosaic fluid? The term mosaic is normally a grouping of elements that differ in terms of size, shape, color, etc. So you group things together, and all those things are different terms of size, shape, and the color, etc. And the whole grouping will be referred to as a mosaic. A mosaic fluid, why? A mosaic fluid because the elements of the plasma membrane, that will say mainly phospholipids and proteins, are not static. they are always in immersion. It's like they are flowing, but without leaving the membrane. And in order to avoid that, the membrane may fall apart. Cytoplasm. The cytoplasm is a semi-fluid matrix that fills the interior of the cell. Already, if we look at this term, cyto, it means a cell, and plasm, it means a liquid. So simply, cytoplasm can be taken as the liquid of a cell. Although the cytoplasm is an aqueous medium, it is more like jelly than water due to its high concentration of organic molecules such as sugars, amino acids, proteins, and inorganic ones, minerals or electrolytes or salts, that the cell uses to carry out its everyday activities. The cytoplasm is a jelly-like substance. It is viscous, it is resistant in terms of flowing because it is highly concentrated in different molecules which are listed here. Among the organic molecules are a variety of enzymes which speed up the many types of chemical reactions involved in metabolism. Enzymes, already which are proteins, are highly required in all activities taking place in the cytoplasm. That is metabolism. And here, once again, remember that metabolism consists of anabolism and a catabolism, and all these reactions are important to sustain life. Note: the part of the cytoplasm that contains organic molecules and ions in solution is called the cytosol. Cytosol. The suffix sol there stands for solution. So cytosol is simply taken as the solution of a cell. A solution of the cell because it indicates the liquidic portion of the cytoplasm in which different organic molecules and ions are dissolved. Second, structures that are suspended in the cytosol, they assume different functions, and these ones are known as organelles. Examples are provided here. We have endoplasmic reticulum, Golgi apparatus, vacuoles, chloroplast, mitochondria, ribosomes, lysosomes, etc. Now we are going to talk about organelles, and here we begin with endoplasmic reticulum. The endoplasmic reticulum, consisting of a complicated system of membranous channels and circuits, is physically continuous with nuclear envelope or nuclear membrane. First of all, the term reticulum means a web, a network. A network of what? A network of channels and circuits. And those channels and circuits are found within the plasma of a cell. That's the reason why here there is the use of the term endoplasmic. A network of channels and circuits, reticulum, found within the plasma of a cell, endoplasmic. The endoplasmic reticulum is of two types. There is the rough endoplasmic reticulum and the smooth endoplasmic reticulum. And these both types are different in terms of structure and function, as we are going to deal with it below. The rough endoplasmic reticulum, first of all, its surface is covered with ribosomes, while the smooth endoplasmic reticulum does not present ribosomes on its surface. That's the reason why one is rough with a surface covered with ribosomes, another one is smooth because ribosomes are absent on its surface. Inside its lumen, the rough endoplasmic reticulum contains enzymes that can add carbohydrate chains to proteins, and then these proteins are called glycoproteins. Remember that the rough endoplasmic reticulum is covered with ribosomes. Then these ribosomes are going to synthesize proteins that will be transported into the lumen of the rough endoplasmic reticulum. Here the term lumen means a free space of a tube or a channel or a sac, etc. Once now proteins reach the lumen of the rough endoplasmic reticulum, they can be processed. The processing of organic molecules consists in adding or removing any part in order to assume any biological importance. Smooth endoplasmic reticulum. Here you should remember that the term smooth is used because this kind of endoplasmic reticulum presents a surface without ribosomes. Now, certain organs contain an abundance of smooth endoplasmic reticulum, and its function depends on the organ. Let's say the importance of the smooth endoplasmic reticulum will depend on the function of the organ containing it. Some organs, smooth endoplasmic reticulum is associated with the production of lipids. In testes, for example, where it is used for the production of testosterone, which is a steroid hormone. And in the liver, the smooth endoplasmic reticulum will help to detoxify drugs, poisons, and the other harmful chemicals that we can take in either by drinking, eating, breathing, etc. So here you see that the role or the function of the smooth endoplasmic reticulum in testes is different from its role in the liver. The smooth endoplasmic reticulum of the liver increases in quantity when a person consumes, for example, alcohol on a regular basis. This means that your liver is going to adapt to the consumption of alcohol when you take it on a regular basis. How? By increasing the quantity or the amount of smooth endoplasmic reticulum. So this explains why two people will take the same amount of alcohol. One is drunk, another remains sober. This is explained by their difference in terms of the amount of smooth endoplasmic reticula that are present in their liver cells or hepatic cells. Let us note that regardless of their difference in terms of their functions, both rough and smooth endoplasmic reticulum form vesicles that transport molecules to other parts of the cell, notably the Golgi apparatus. we'll see how the transportation of substances within a cell is principally assumed by the coordination between the endoplasmic reticulum and the Golgi apparatus. Lysosomes. Lysosomes are membrane-bounded vesicles produced by the Golgi apparatus. This means that we have the Golgi apparatus as an organelle that gives rise to another organelle that will be known as a lysosome. Lysosomes have a very low pH and store powerful hydrolytic-digestive enzymes in inactive state. Lysosomes contain enzymes that will be used by a cell to deal with the problem of microbes and the problem of organelles, especially when they are non-functional, old or damaged. And it's very important to note that the enzymes in lysosomes should be kept in their inactive state in order to avoid accidents or troubles that may occur when, for example, lysosomes burst by accident. Materials can be brought into a cell by a vesicle or a vacuole formation at the plasma membrane. When a lysosome fuses with either, the lysosomal enzymes are activated and digest the material into simpler subunits that then enter the cytoplasm. What does it mean? A vesicle or a vacuole is going to be formed by the plasma membrane. And this vesicle or vacuole will surround a particle to be taken into the cytoplasm. Then the vesicle or vacuole will have to fuse with the lysosome that contains hydrolytic enzymes to break down the particle into very simple subunits that can be easily absorbed by the cytoplasm. Some white blood cells defend the body by engulfing bacteria that are then enclosed within vacuoles. When lysosomes fuse with these vacuoles, the bacteria are digested. You know that white blood cells defend the body against infections, and this is done using different means. And among them, there is the phagocytosis of microbes, especially bacteria. So once a bacterium is packaged into a vacuole, that vacuole will have to fuse with a lysosome containing hydrolytic enzymes that will be used to break down or destroy the microbe, a bacterium in this case. After the molecules of a bacterium are broken down into very simple subunits, these ones will be absorbed into the cytoplasm. Then, sometimes a small amount of residue is left and then it is ejected from the cell at the plasma membrane. The digestion takes place. Hydrolytic enzymes of the lysosome break down a microbe. Some small subunits are absorbed into the cytoplasm, and the leftover is going to be remaining in what we call the residual body. It is that residual body, which is normally also a sac or a vesicle, is going to approach the plasma membrane and fuse with it in order to discharge its content out of the cell. That will be now the exocytosis.